So last time we chatted, I talked with you guys a little bit about the different options for pyruvate once pyruvate was made during the process of glycolysis. So we talked really briefly about the fact that some organisms like yeast can undergo fermentation, which is going to take the pyruvate molecule, remove a carboxylate group to produce alcohol, and have ethanol remaining as the final products of pyruvate. In the skeletal muscle, I talked about the fact that pyruvate can be used to make lactate or lactic acid. Today, we're going to talk about what happens with pyruvate under the ideal conditions where oxygen is available and we can get as much energy out of those original bonds of glucose as we possibly can. So the initial step that's going to get us to that place where we can break down pyruvate completely into carbon dioxide and water is going to be a linker step that leads to the production of acetyl-CoA. So under aerobic conditions, pyruvate is decarboxylated. Now this is a term that you're going to see a couple of different times today, and I just want to highlight the word parts here to make sure that you understand what this word is trying to relay to you. So D is the prefix that is without or devoid or indicates the removal of something. And carboxyl is referring to the carboxyl group. So remember that the carboxyl group is what we have with carboxylic acids. It's a carbon atom with a double bond O and an OH. Now in most cases the H is donated as an acid or because this has acidic properties, and that leaves you with an O minus. So decarboxylation, or something that is decarboxylated, indicates that it's removing this group, and it's going to remove that group to produce carbon dioxide. Remember that carbon dioxide is a carbon atom with a double bond to both of these two oxygens. So it's a C in the middle with a double bond O to the right and a double bond O to the left. Now that's going to take our 3-carbon pyruvate product and reduce us down to 2 carbons. These 2 carbons here that are complexed to coenzyme A to yield acetyl-CoA. So this group is an acetyl group. So what this means is that our pyruvate molecule has now produced for us the first of 3 carbon dioxide molecules that will arise because of the complete oxidation of pyruvate. Now, as this CO2 is produced, this CO2 and this decarboxylation reaction is an oxidation reaction because we're increasing the carbon-oxygen bonds for this carbon. In pyruvate, the carbon has 1 double bond O and a single bond O. So it's being oxidized by the production of a second bond right here to produce carbon dioxide. So we already know that every time something is oxidized, something else has to be reduced. And that is where NAD and NADH come into play. So energy is being carried, energies and electrons are being carried by our NADH molecule. So I'm going to divide the citric acid cycle into 2 sections for you. The first 4 reactions are going to be the focus of this particular little talk. And we're going to focus on the fact that in these first 4 reactions, we produce 2 more molecules of carbon dioxide. So the first half of the citric acid cycle, so the first half over here, is focusing on the production of carbon dioxide. The rest of the citric acid cycle is going to be to get back to the starting place that we're going to talk about with reaction 1. So the bottom line for the citric acid cycle is that 2 carbons of acetyl-CoA, which would have again originally been a part of our glucose molecule at the very beginning, these 2 carbon atoms are added onto a 4-carbon molecule to make the 6-carbon molecule citrate. We're going to go from citrate to a 5-carbon molecule alpha-ketoglutarate. We're going to do that by the loss or the production of carbon dioxide. And then we're going to go from a 5-carbon molecule to a 4-carbon molecule Again, we're going to do that by the release of the last carbon dioxide from our pyruvate molecule. So the citric acid cycle begins with acetyl-CoA. And it's only going to operate under conditions where oxygen is present. Now, you're not going to see oxygen directly in the citric acid cycle, but I will point out to you later where that oxygen comes into play. So our acetyl-CoA enzyme A is carrying for us the 2 carbons that came directly from pyruvate. Now, it's going to be donating 2 carbons into the citric acid cycle, and by the end of the citric acid cycle, we will have produced 2 more molecules of carbon dioxide. Now, when we produce carbon dioxide, that is going to be an oxidation reaction. So at the same time we are producing carbon dioxide, we are also producing high energy molecules like NADH and FADH2. Now, the NADH molecules will always be derived in the same steps where our carbon dioxide molecules arise. And then we're going to also generate in the second half of the cycle FADH2 and ATP. So what we're looking at right now is going to be the cycle right here. So the acetyl-CoA brings 2 carbons into our citric acid cycle. The products of our citric acid cycle are carbon dioxide and high energy molecules that will ultimately be used in the electron transport chain to produce ATP. Now, the electron transport chain will be discussed in the last part of this chapter. But for right now, we're going to think about the citric acid cycle, production of carbon dioxide, and in this first half, the production of NADH. So the acetyl group brings in 2 carbons to generate a 6-carbon molecule for us. We'll then have oxidation reactions occurring where we remove carbon dioxide and then eventually revert our 4-carbon molecule back to oxaloacetate so that the cycle can be repeated again. So as we go through this process, I want you guys to be on the lookout for oxidation and reduction reactions that we've already seen before. So the initial ones we're going to see are going to be the conversion of hydroxyl groups to carbonyl groups, like we saw with the conversion of alcohols to aldehydes and ketones. We're going to see that kind of reaction. In the second half of the cycle, we're going to see single bond carbon-carbon bonds being converted into carbon-carbon double bonds. So think about that, and hopefully this will allow you to keep straight whether the molecule is being oxidized or whether the molecule is being reduced. And again, I'll point these out to you as we go along. So these are the reactions of the citric acid cycle. Don't allow yourself to freak out because of how complicated it looks. I'm going to go through each of these reactions and point out the key features and things to look out for. Again, you're not going to have to draw these molecules, but I want you to be able to look at one molecule and the next molecule in the reaction and be able to understand or articulate what is happening through the course of that reaction. So the first reaction is the addition of our 2-carbon unit from acetyl-CoA to our molecule oxaloacetate. Oxaloacetate is a 4-carbon molecule that has a carboxyl group at each end. Now the addition of the acetyl coenzyme, or the acetyl-CoA, the addition of the acetyl group from acetyl-CoA is going to result in the formation of the molecule citrate. Now this molecule has now 3 carboxyl groups. And remember that 2 of these carboxyl groups are going to come off in the form of carbon dioxide molecules. So the first reaction is taking a 4-carbon molecule and a 2-carbon molecule and making our 6-carbon molecule. Now citrate is a complex molecule, as you can see, and if you look right here you can see that it has a tertiary hydroxyl group. Now you guys already know from back in our study of alcohols that a tertiary alcohol can't be oxidized. And so the citrate molecule has to be rearranged. And it's going to be rearranged by the movement of the OH from a tertiary OH to a secondary OH. And again, an enzyme is going to do the steps of this reaction. The initial step is to remove or dehydrate our molecule to make a double bond O. So again, we're going to take away an oxygen and 2 hydrogens to form water, but then we add back a water in the very next step to produce the secondary alcohol that we want. So we're converting a tertiary group to a secondary group that can be oxidized in the next step. So we're just rearranging or isomerizing the citrate molecule to isocitrate. And that's what's depicted here. So all we're doing right here is changing the location of where the hydroxyl group is located. Because as a tertiary OH, it can't be oxidized, and as a secondary OH, it can. So isocitrate is going to undergo there are going to be a couple of key features to think about in this reaction. The first is the first removal of a carbon dioxide, so the first decarboxylation reaction. So we take a carboxyl group, it's going to be the carboxyl group in the middle right here. The carboxyl group is going to be removed to produce carbon dioxide. Now, as you guys know, anytime we have oxidation, we have to have reduction going on as well. So we're going to talk about oxidation in the next portion by focusing right here on the secondary hydroxyl group that we generated in the previous step. So this secondary hydroxyl group is going to undergo oxidation to produce a double bond O. So again, you guys already know that oxidizing an alcohol produces a ketone, and that's exactly what's happening here. Now when this happens, there's going to be the release of the hydrogens that you see here in blue. Now these hydrogens and 2 electrons are going to be donated to an NAD to produce NADH. So again, the oxidation that's occurring here at the OH to make our double bond O has to be partnered with a reduction reaction, which is going to give rise to our NADH. So the NAD accepts 2 electrons and a proton to become NADH, and the other hydrogen can basically replace or provide the 4th bond to the middle carbon right here. So both of these, the oxidation reaction and the reduction reaction, have to go hand in hand. So again, I told you guys earlier on that everywhere we produce a carbon dioxide, we also produce an NADH. So if you know where one is produced, then you automatically know where the other is produced. And you can see where the carbon dioxides are made because we're going from a 6 carbon molecule over here to a 5 carbon molecule over here. Okay? Now we're going to have a very similar kind of reaction at reaction 4, the very next reaction in line. Again, we have a decarboxylation reaction. We're going to take the carboxylate group at the end, and we're going to generate carbon dioxide by the removal of that group. Now again, we're going to have the production of 2 protons and 2 electrons so that this oxidation step is going to be partnered with the reduction of NAD to NADH. So the NAD is going to take 2 electrons and a hydrogen to make NADH. Okay? So again, if you see where carbon dioxide is made, you automatically know that an NADH is going to be made at the same time. So that reaction is depicted here. Again, we've got our 5 carbon molecule, alpha ketoglutarate. The alpha ketoglutarate is going to lose a carbon and 2 oxygens in the form of carbon dioxide. And when it does that, we're going to be left with a 4-carbon chain. Now the 4-carbon chain is temporarily attached to a coenzyme A molecule, and we'll talk about that whenever we go through the rest of the citric acid cycle. But so far, we've already gone through 4 reactions, and we've seen the production of 2 NADH molecules and the removal of 2 carbon atoms in the form of carbon dioxide. So one right here when we go from isocitrate to alpha-ketoglutarate, and then another molecule of carbon dioxide when we go from alpha-ketoglutarate to succinyl-CoA. Now the rest of the citric acid cycle is going to be taking our 4-carbon chain and reverting it back to the original molecule, acetyl-CoA, so that the cycle can be repeated. But for now, take some time to understand the process and what's happening in reactions 1 through 4 of the citric acid cycle.